Hello, classmates! May I ask if everyone has played with Lego? Lego can be assembled into all kinds of machinery. There are all kinds of functions. So, does everyone know how many kinds of Lego blocks are needed to assemble these Lego machines? In contrast, only 20 kinds of amino acids make up the proteins in our human body. But they are pieced together to form our running legs, a beating heart and a thinking mind. How can all this be achieved? We need to start with the relationship between the structure and function of proteins. We will focus on learning this lesson. 4 key relationships between the primary structure and function of proteins. Secondly, we also need to understand the relationship between the spatial structure and function of proteins. Does everyone still remember the primary structure of proteins? It is the sequence in which the amino acids are arranged. If the amino acids accidentally line up in the wrong order, it's just like using the wrong blocks when building with Lego. What will happen then? Let's take an example. Sickle cell anemia. The patient's red blood cells will turn from small discs into small sickles. It causes a series of symptoms such as hemolysis. This disease is caused by a misalignment of one amino acid in the beta subunit of hemoglobin. That is, glutamic acid No6 at the N-terminal is replaced by valine. The mistake of just one amino acid led to such serious consequences. This leads to the 4 key points that we need to master in this class. The primary structure of a protein is the basis of its spatial structure. Altering the primary structure of a protein can directly affect its function. Third, if the primary structures of proteins are similar, their functions will be similar. Fourth, the primary structure of homologous proteins contains information about biological evolution. Let's take a look at sickle cell anemia again. After glutamic acid is replaced by valine. Because the R group of valine is non-polar, and it is uncharged. Therefore, the repulsive force between hemoglobin is reduced. Promote the coagulation of hemoglobin and turn red blood cells into sickle-shaped ones. From this, we can see the primary structure of a protein is the basis of its spatial structure. Let's look at another example. Bovine pancreatic ribonuclease can catalyze the hydrolysis of RNA. Its primary structure contains 4 disulfide bonds. 4 disulfide bonds polymerize the peptide chain into clusters. When we use mercaptoethanol and urea to break its disulfide bond, the peptide chain then becomes a long strip. Its catalytic effect is gone either. After we removed mercaptoethanol and urea, disulfide bonds can be restored. The peptide chains will reaggregate into clusters. Its catalytic function will also be fully restored. Here we can see it again. The primary structure of a protein is the basis of its spatial structure. In addition, we can also find out altering the primary structure of a protein can directly affect its function. There are no disulfide bonds. It cannot form a group. The catalytic function naturally doesn't exist either. Because the primary structure is of great significance to the function of proteins. Therefore, the situation where amino acid dislocations occur is naturally not allowed. If we compare a protein to a key, the function of protein is like that of a lock. In order to unlock, the key teeth must not be changed. The primary structure at this position is what we call a conservative residue. The position of the key handle, however, allows for certain changes, what we call variable residues. Then it can be seen. As long as the amino acid arrangement of the key teeth is similar, those 2 keys must be used to open the same lock. That is to say, the primary structures of proteins are similar, its functions are also the same. For example, bovine insulin. It has the same chemical structure as the natural insulin molecule. And it has complete biological activity. It is very similar to the conserved residues of human insulin. So bovine insulin can also have a hypoglycemic effect on the human body. When it comes to bovine insulin, it is the result of nearly 7 years of scientific research efforts by Chinese scientists. It was first fully synthesized artificially in the world in September 1965. This achievement has caused a huge response internationally. It is a model of cutting-edge research in our country and a major achievement in the basic research of natural science in China. This also marks the beginning of the era of artificially synthesized proteins. In addition, the keys in our bodies may have been passed down for hundreds of millions of years. In the endless passage of time, although the key handle has changed beyond recognition, however, the key teeth still have few changes. It contains information about biological evolution. I've finished learning this part. Let's do a little test. The following 4 peptide chains. Which 2 might have similar spatial structures? What about the functions and kinship? Except for the primary structure, the spatial structure of proteins also directly determines their functions. First of all, the conformation of a protein determines its properties and functions. For example, long fibrin. It is most suitable for forming muscles and hair strands. But it's for running around in the body to participate. All kinds of chemical reactions are just too inflexible. In contrast, the round spherical protein cannot be piled up into a solid shape. But they have all kinds of active centers. It can help us regulate various physiological activities throughout the body. In addition, our proteins can also be assembled into transformers just like Lego. For instance, the hemoglobin we mentioned earlier is a master at transforming. After hemoglobin binds to oxygen, the affinity of its 4 subunits has weakened. Its 4-level structure can shift from a tense state to a relaxed state. This deformation is subject to the partial pressure of carbon dioxide. Adjustment of factors such as pH value. It can help hemoglobin carry and release oxygen. Secondly, some proteins have specific structures. It can specifically bind to certain structural molecules. These molecules are also called ligands. After binding the ligand, these proteins will also deform. This then triggers more biological effects. Finally, here's a thought-provoking question for everyone after class. Take hemoglobin and myoglobin as examples. How does the spatial structure of proteins determine their functions? And analyze the significance of this structure-function relationship in biology. After the learning in this class, I believe everyone has already understood the relationship between the structure and function of proteins. Now let's summarize. Horizontal inscription. Level 1 is the foundation. The ancestral primary structure defines space and function. Variable spatial structures determine functions and ligands. Dear classmates, that's all for this section. Goodbye.